Hi guys, so we already have seen transformation and conjugation. Now the third and the last method of horizontal gene exchange in bacteria is transduction. Okay, now for transduction to occur, we have to familiarize ourselves with a special kind of virus called as bacteriophage, or it is simply called as phage. It is nothing but a virus which attacks a bacterium. Okay, this is how it looks like. All right, so basically it has got these parts, the head, right, and the head is going to have the genetic material. Remember, viruses have got either DNA or RNA, never both. So in this diagram we have taken DNA as the genetic material of this bacteriophage or simply phage. All right, then there's a collar, the tail, the black portion around, all around over here is nothing but the protein, right, and we have got what is known as the base plate and the long tail fibers. It is with the help of these long tail fibers that the bacteriophage is going to impinge upon or attach onto the bacterial cell, right. So it somewhat looks like a spider. All right, this is just a cartoonish representation to emphasize upon the structure of bacteriophage. All right, this is the actual picture, the electron microscopic picture of bacteriophages. You can see this is a giant bacterium, and you can see all these are what? These are bacteriophages. Okay, this one, everything, all these are bacteriophages. All right, so let us see what a bacteriophage will do when it attacks a bacterial cell. Okay, now let me zoom in. Now let us say this is a phage and this is a bacterial cell, B for bacteria, okay, P for phage. Okay, now with the help of its tail fibers, it is going to attach onto the bacterial surface receptors, right. The bacteria will be expressing certain receptors on its surface to which the tail fibers are going to attach, and this is the bacterial chromosome. Okay, this curved thing. Next, what will happen? Just like when we are giving injection to somebody, only the contents of the syringe, only the liquid goes inside, right. Similarly over here you can consider this bacteriophage as a loaded syringe, and only the DNA is going to enter this bacterial cell, and everything else will be outside, will remain outside. Okay, so the bacteriophage is going to just inject its genome, its DNA into this bacterial cell. All right, and once this happens, an important change is going to happen in the bacterial cell. I want you to compare and contrast figures 1 and 2 with regard to the bacterial chromosome. In figure 1, the bacterial chromosome is intact. Now look at figure 2. The moment the phage DNA, which is shown in blue color, the moment it enters this bacterial cell, the bacterial chromosome undergoes disintegration. This is very important. Okay, now next what happens, as as is the case with viruses, that they are very poor in the sense that they have got information or genetic material to to replicate their genome and to form their proteins like capsid, peplomar, etc. Similarly in phages also, because they are viruses, they are not able to multiply independently. So when this DNA enters, the phage DNA enters inside this bacterial cell, it is going to manipulate the bacteria to synthesize its own genome and proteins, right. Even though bacteria don't have, bacteria are not going to like it, the entry of this phage DNA into the bacterial cell, right, but the bacteria cannot do much about it. The phage is going to take over, all right, and the phage is going to synthesize its components, that is it is going to replicate its genome and it is going to synthesize its own proteins, right. So individually these genomes and proteins now have to come together, that is they have to assemble together to form the daughter phage particles. Now imagine there is a factory, which is engaged in the manufacturing of robots, right. Let's say on ground floor the head of the robot is being manufactured, on the first floor the trunk and the limbs on the second floor. Now from all the floors, all these components have to be assembled together to form the intact robot, and that is what is done during assembly. that is the replicated genome and the proteins have to come together to form the phage particles. Okay, so during assembly what is happening, the head, tail and tail fibers, they come together to assemble the phage, right. So once the daughter phages have been formed, they are going to be released by lysis of the bacterial cell, all right. And then these phages are going to then attack other bacterial cells, all right. So this is how the phage and the bacterial interaction is going to be, right. Now I am going to tell you about some error which might happen in case of certain phages when they attack bacteria. Guys, mistakes only are not committed by humans, but such tiny fellows, I mean viruses are the tiniest of all, right. These fellows can also do mistakes in their life, right, and then the bacterium might have to face the music, right. Let us see. We are going to see what is known as generalized transduction. Okay, what does the transduction by the way mean? Something is being changed or transduced. Okay, now we are actually going to see what exactly do we mean by this. So we have got this cell 1, this is a bacterial cell shown in blue color. So this is cell 1 and we have got this phage. I want you all to carefully appreciate the colors. The phage genome is purple in color, right. So initially what happens, the phage adsorbs onto the specific cellular receptors and injects its DNA into the cell, just like we saw in the previous diagram. So this is the phage DNA which is purple in color, right. So next what happens, the early transcription and translation of phage DNA occurs, right, to convert the cell into a phage factory and to replicate phage DNA. This already I have explained in the previous diagram, right. And one more important change I want you all to notice is, look at this, the bacterial chromosome over here was a perfect circle, light blue colored circle. The moment the phage DNA enters, the bacterial chromosome undergoes fragmentation, right. So remember, during viral replication, early and late proteins are synthesized. Early proteins are nothing but the enzymatic proteins and the late proteins are structural proteins. Okay, so suppose this purple circle is the phage DNA, and then the early mRNAs are, this is the early mRNA, the green one, and these are the early proteins. Okay, now after replication of the DNA, the phage structural proteins are replicated at a higher rate. Okay, so what is basically happening, the phage DNA entered, then the early mRNA was synthesized, and then early proteins were made, which are nothing but enzymes. These enzymes are going to be required for formation of further proteins, that is late proteins, and for genomic replication also, right. So what happens, look at this, now you can see this bacterial cell is now looking like a fish market, with so many different types of phage proteins and phage genome etc. are present, right. Just remember that example or analogy of a factory manufacturing the robots. So the different components of the phage are being synthesized now in the bacterial cell, and then there is going to be assembly of the phage. Okay, assembly is also known as morphogenesis. Morpho means morphology, genesis means to make, right. So once these daughter phages are assembled, they are going to be released by cell lysis. So you might be wondering why I have put up a separate diagram to tell the same thing which I told previously. There's a reason, guys. As I mentioned in the beginning, you have to emphasize, see the colors carefully in this diagram. Now let me label the daughter phages in this figure as 1, 2, 3, 4, 5. Are these 5 different from each other or the same? Now guys, phage number 1, if you notice carefully, its genome is blue in color, it is not purple, whereas in 2, 3, 4, 5, purple DNA, alright. Why is this blue color DNA present in daughter phage number 1? This blue DNA, guys, is nothing but, look at this, let me use red color to encircle this, look at this. One of the broken fragments of the bacterial chromosome has been falsely, erroneously picked up or assembled during the assembly of this phage number 1. Okay, so what has happened, this one phage, that is phage number 1, which I have marked in green color, this one phage has incorporated a piece of bacterial DNA by mistake. It is called as a transducing phage. Alright, now guys, out of so many different, look at this again, out of so many different bacterial chromosomal fragments, any fragment can be picked up can be erroneously picked up during assembly of the phage. So we say that any bacterial gene by mistake can become can enter the phage. So it is generalized. It is not that a specific bacterial gene is only going to enter the phage during the assembly. Okay, that is why this process is known as generalized transduction. Okay, so it's a transducing phage. It's a generalized transducing phage because every bacterial gene has had an equal chance of being incorporated while it when it infects another similar cell. Now let us see over here. Let me highlight this in yellow color. Now when this phage, which is having the bacterial fragment, the bacterial DNA fragment, let us say it is attacking another bacterial cell, cell number 2. So when it infects another similar cell, when it affects another similar cell, it takes bacterial genes from cell 1 to cell 2, okay, creating the opportunity for new combinations to occur. Now and let me underline the word similar. Okay, so we are saying that when this phage infects another similar cell, the word similar is important over here because let's say cell 2 is similar. Similar in what sense? Similar in terms of homology of genes. Alright, so this phage has now injected its DNA, which is actually the DNA of the bacteria. So bacterial DNA from cell 1, remember we started with cell 1, I named this yellow bacterial cell as cell 1. Okay, so this cell number 2 has got this this green protein, which I told you while talking about transformation. This is rec A protein or recombinase, and cell 2 is a cell which is somewhat similar to cell 1 in the sense that its genes are homologous to cell 1. Okay, so when rec A gene product, that is rec A protein, is present and both the cells are similar, bacterial cell 1, bacterial cell 2 are similar to each other, then it means that the genes are homologous, then homologous recombination can take place. So new recombinations are possible if homologous recombination produces an allelic exchange. Okay, so this is how, through the agency of the phage, what has happened. From bacterial cell 1, some genes have transferred into bacterial cell 2, and as I explained, it's just a matter of chance or randomness that any bacterial gene could have been erroneously picked up by the phage and entered cell 2. That is why it is known as generalized transduction. Alright, so this is another figure of generalized transduction. This is the phage, the blue one. Alright, it adsorbs onto the bacterial cell and injects the DNA, which is shown in red color. Then this is the green, what you are seeing in green is the bacterial DNA, right? It gets broken down. Then all these are fragments, are the components of the phage, the phage genome and the phage proteins, and then assembly occurs and the cell lyses. And look at this. This phage has its genome is green in color, and again it means that it erroneously or by mistake has packed up or assembled in itself a fragment of the bacterial genome, which is green in color. Okay, now let us understand another phenomenon called as lysogeny. Okay, what is lysogeny? Lysogeny is a phenomenon which has been well studied in Escherichia coli, and let us see what happens. Now let's say, see guys, there is a special kind of phage called as lambda phage, which is very well studied, right? So this lambda phage is shown over here in purple color, this one. So lambda DNA, this is the lambda phage, by the way, the lambda phage, and this yellow colored bacterium is E. coli. The lambda phage has adsorbed onto the E. coli and has injected its DNA, which has become circularized. So this purple circle is nothing but the phage DNA, right? And there is a protein called as lambda repressor. Okay, the bacterial cell is making a protein called as lambda repressor. Okay, as the name implies, this protein is going to repress or tone down or cut down or stop the activity of the lambda phage. So if this lambda repressor, which is shown in this or as this orange triangle, if this is made quickly enough, what happens? The phage will not multiply now in this bacterial cell. So active phage production is inhibited. As the name implies, lambda repressor, the lambda should be repressed or suppressed. Okay, now I want you all to focus on two important genes in this E. coli. One is for the synthesis of biotin, that is bio plus, and the other one is for galactose metabolism, that is gal plus. Okay, so what happens, the lambda repressor protein is not anyways going to allow the lambda phage to multiply. So lambda does what? It is bored. So lambda DNA inserts itself into the bacterial chromosome, but specially and only between the bio and the gal genes. Okay, so it is very very picky about the place where it is going to go get inserted. Okay, like we are picky about the movies we watch, the restaurants we go to, the places we hang out at, like that. So now this lambda phage wants to hang out, but it wants to get itself incorporated only between these two genes, gal plus and bio plus. Okay, so as we, this is what we discussed in the beginning when I was talking about site specific recombination. Okay, so this is the same thing, guys. Look at this. You can see this part this is looking like a door, the sky blue color region in this bacterial chromosome, and look at the phage DNA also. It also has got this, let me encircle this part. Okay, this is also looking like a door. So basically at these sites, these DNA pieces are going to open up and then bind to each other, just like over here what has happened, right? So now what do we call this E. coli? Now we call now what do we call this phage, by the way? Lysogenized lambda. Okay, this is called as lysogeny. Okay, so the lambda phage production is repressed because of the lambda repressor protein which is present in the bacterial cell, but the lambda DNA, because it has become now part and parcel of the bacterial DNA, it also replicates every time the E. coli DNA replicates. Okay, so no problem is happening right now. The bacterium is at peace, the phage is also happily, the DNA is multiplying and all. But let us see what happens when this lambda repressor is suppose damaged because of some reason. Okay, so now let us say that the lambda repressor gets damaged. Now as the name says, lambda repressor, when this repressor is not there, so it is not going to repress lambda, so lambda phage multiplication should occur. Okay, now for lambda phage replication to occur, what happens, the lambda phage whose genome in which I have shown in purple color, which had got itself integrated into the bacterial chromosome, now it has to come out to multiply, right? That is, it has to excise itself. Now if the excision is normal excision means it is going to not commit any crime in the sense that it is not going to steal any genes from the bacterial chromosome, right? So it is simply coming out, right, like this, and again then it will multiply, and the daughter phages, the lambda phages are going to be produced. Okay, so this happens in case of a normal excision. Normal excision meaning when the lambda phage DNA decides to come out of the bacterial chromosome, it is not going to take along any of the bacterial genes. But as I said, mistakes happen, not just we commit mistakes, but phages also can do the same. Now let us say what if there is an excision error in a lysogenic phase. Okay, so excision error means that while coming out, while getting excised from the bacterial chromosome, the lambda phage picks up a bacterial gene into its own genome. Now because it got inserted between the bio and the gal gene, these genes are next to each other. I mean, these genes are on either side of the phage DNA. So when this phage comes out, it can take either of these genes. You might think why it can't carry both these genes. Guys, it is going to pick up either the bio gene or the gal gene, because if both genes are picked up then the length of the DNA segment is going to be too long, okay, which is not permitted. So while coming out, let us say it has, so see, I want you all to focus on this part. Can you see? So this is excision is happening, but during excision, this sky blue colored part represents the bacterial chromosome or the bacterial gene. And what gene it has incorporated? Galactose metabolism, that is gal plus gene. Okay, so now this phage is going to multiply, and this phage is now transformed. Why? Because it is now going to have or possess the bacterial gene also. All right, now let us say so now this is nothing but the lambda D gal plus, that is D galactose plus phage. Now let us see what happens if such a phage which is having some bacterial genes, it is going to attack some bacterial cell. In other words, let us see what does specialized transduction mean. Now, specialized transduction means that when this phage is going to attack this bacterial cell, the yellow one, now it has got now it has injected the genome which it was carrying into this bacterial cell, and that genome had not just the purple gene but the blue color gene also. The blue color gene was the gal plus gene. All right. Now this bacterium itself is gal negative, right? It doesn't have gene for galactose metabolism, right? But this bacterium has got rec A gene or the rec A protein. So if the cell is rec A plus, that which means that the recombinase A protein is being produced, then the gal gene may be stabilized by homologous recombination. Okay, so gal plus, gal minus, these are homologous genes, and if this yellow bacterial cell has got recombinase A protein, then exchange is possible, and the gal minus of the present in the bacterial chromosome will go out and gal plus will be incorporated. This is nothing but homologous recombination. Okay, homologous recombination, right? This is known as specialized transduction because only special genes, only specific genes like gal plus or biotin plus genes only can be transferred from one bacterial cell to another in this way, precisely because the lambda phage was very picky about the place where it wants to insert itself into the bacterial chromosome. Okay, again this is another representation of specialized transduction. So this is the bacterial DNA, this is the phage, and this is the phage DNA. Okay, so the phage DNA gets incorporated into the bacterial DNA, right? The bacterial DNA is in zigzag fashion, and this one like this is the conformation or configuration of the phage DNA. Okay, like this. Then suppose there is aberrant or abnormal accession of the phage DNA, right? Remember the phage DNA got incorporated between the gal and the bio genes. So next what happens during abnormal accession, this phage has taken along biotin gene. Okay, now this phage multiplies, right? This phage will then be released, and then it can attack another bacterial cell and transfer the bio gene. Okay, now that we have covered transformation, transduction and conjugation, let us see what are the important differences between these three. Is cell to cell contact required? It is required only in conjugation, not in transformation or transduction. Remember, C for contact, C for conjugation. We saw how conjugation tube or bridge is formed between two cells during two bacterial cells during conjugation. Now, does it require an antecedent phage infection? Phage I have spoken to you about only during transduction, not in conjugation or transformation. Is competency required? Where was I explaining the meaning of competency during transformation? Remember, I told you that the bacterial cell will become competent enough to bind to the dead DNA pieces when it expresses certain proteins on its surface, which are going to be just like Fevicol or Fevistick, facilitating the transfer or attachment of the dead DNA. So that is seen in transformation, not in conjugation or transduction. Is naked or free DNA involved? Yes, it is involved in transformation, not in conjugation or transduction. Is recombination required to stabilize new genes? Yes, in transformation it is required, just like we saw homologous recombination. In transduction also we have seen examples right, of homologous recombination. In conjugation, recombination is not required for F plus and F minus crossing. All right, only the plasmid is being donated. There is no change happening in the bacterial chromosome gene, but it is required, but the recombination is required, as we saw in the crossing between HFR and F minus cell. Okay, then comparison of generalized and specialized transduction. The mechanism of generalized transduction is error in the assembly, as we saw. In specialized, it is error of the accession. Okay, so it requires stable insertion of the prophage DNA. Okay, lysogeny. All right, and what genes may be transferred? As I explained, in generalized transduction any gene can be transferred, and in specialized transduction only the genes next to the insertion site, as we saw in case of lambda phage getting incorporated between the biotin and galactose genes, only these genes can be transferred. So guys, that's about bacterial genetics.